All right, so welcome to week 1 lecture of microbiology. Today's lecture will focus on an introduction to the field of study and will cover primarily chapter 1 in your textbook. So I hope by the end of this week 1 lecture, all of you will have gained a greater appreciation for the microbial world. Unfortunately, there's this awful stigma associated with microscopic life, and that stigma is that all microbes are bad. All microbes are germs that cause disease, and you should clear all microbes from your environment. Use antimicrobial or antibacterial soaps to ensure that there are no microbes anywhere in your surroundings. And I think this cartoon illustrates this negative stigma. You have a student that says, it says here we have microorganisms living in our noses. I'm totally grossed out. So this cartoon illustrates the notoriously negative view of the microbial world, and I am here to hopefully convince all of you that the majority of microbes are actually our friends and not our enemies. We know that microbes are ubiquitous. They are everywhere. They are all over surfaces in your surrounding environment. They are in the air attached to dust and dander. They are on the order of millions and billions per single gram growing in soil in your backyard. And they are all over the surface of your skin and within the human body. So per square inch, 2 million microbes on human skin. 1 single gram of feces contains 100 billion bacteria. And I just love this third fact here that the human body consists of 100 trillion cells. And of those 100 trillion cells, only 10% of those cells are mammalian eukaryotic cells. The remaining 90% are microbes. So we are all walking around as these human ecosystems individually comprised of 90% in cell number bacteria. And 90% of those cells, which are microbes, together weigh 2 to 6 pounds, which is actually anywhere from 1 to 2 times the size of an adult average human brain. Also, the genetic load within the human ecosystem, there are actually 150 times more microbial genes contributing to the human biology environment. So this is a staggering number here, 150 to 1. So microbes are everywhere, and they're living on the surface of our skin, and they're residing within the human body as well. Now, the ones that live on skin and in the human body are referred to as the normal or the commensal microbiota. These bacteria on the order of potentially billions living in our guts, but as far as the strain or the species type, on the order of 10,000 plus species can exist within a single human body. And they're called normal or commensal because they maintain human health. They are good for us. They are not bad for us. They don't make us sick. We need all of these microbes to maintain our health status. Now, the gut is the primary location within the human body where you will find the most microbes out of that 90% number that I mentioned. So there are billions of microbes in the small and the large intestine. You also find them in the upper digestive system, the stomach, along with other parts of the body, which I'll point out in a couple of slides. But within the gut, all of these microbes are serving a multitude of functions, from protective to structural to metabolic. So this is everything from nutrient competition, producing their own antibiotics, but also developing the immune system. So normal, healthy gut microbes produce the majority of IgA antibodies. This is immunoglobulin A. They also are responsible for metabolizing dietary carcinogens. So for anyone who likes to eat grilled food, you are consuming carcinogens when you eat that food. The normal human epithelial cells may not be able to metabolize some of those toxic compounds, certain microbes can't. So they can actually serve as an anti-cancer function. They also are required to produce a multitude of vitamins like biotin and folate, and the list goes on and on. All of these are, again, healthy. If we take a look at microbes in the stomach as compared to in the lower digestive system, the small and the large intestine, we see that there are some bacteria you find here, 10 to 1 to 10 to the third CFU. This stands for colony forming unit. A colony is basically a group of bacteria derived from one single cell. I will talk about that in more detail later. But you see the numbers are higher here as you go and traverse through the digestive system. Majority you find in the colon. And over the years, we've taken samples of gut microbiota from human colons, and we've profiled those samples, and we figure out what is living in the normal human gut, and we find there are all different types of genre of bacteria, both aerobic and anaerobic. These terms are referring to oxygen requirements for those bacteria. So aerobic, requiring oxygen or liking oxygen. Anaerobic is without. Now, again, I want to stress this, that every single human being, 10 to 1 cell number is made up of microbes over human mammalian cells. So where do you find all of these microbes? Well, within the oral cavity, so within the mouth. The mouth is actually considered one of the most diverse microbial environments. There's over 400 to 600 unique species found in the mouth. Again, all over the surface of skin, every inch is covered with microbes. The nose, the throat, and now we're finding that the lungs are not sterile, so there are even normal microbes that may reside in the human lung. And for sure, we know the gut, the colon, small and large intestine, and also the stomach is highly colonized with microbes. Now, what's really interesting, and I wanted to throw this research finding and piece of data in here to share with you guys, that you can think of the normal human microbiota similar to and as unique as a fingerprint. So what I mean by that is that every single human being has their own unique normal microbiota set or profile. So you are unique as far as the types of microbes that are growing in your nose or in your mouth or on your teeth, your tongue, in your gut, and etc. So if you take a look at the top image here, every single bar is representative of one human sample from a particular site of the human body. And as you can see, they are all different, very diverse as far as the types of microbes that are found in those samples. And interestingly, even though they're very diverse with regards to the types of microbes that live there, there's this consistency in how the metabolism of the environment is functioning as a whole. So there's basically the take-home message here is there are many different ways to establish the same metabolic ecosystem with varying levels of different types of bacteria. But I do believe that the different types of bacteria that are found unique to an individual, you could think of this as the ultimate selfie. So if you were to actually get a picture of all of the microbes that make up who you are, this would put things into perspective, right, as far as who we are as individuals. Now, what I think is so exciting is that all of these microbes that I'm trying to convince you guys are healthy for us, the 90% that makes up the human body, those microbes seem to maintain, contribute to, and determine your health status. So depending upon what's growing in your gut, what's growing in your mouth, on the surface of your skin, can determine how healthy you are. Those microbes can determine how your immune system functions. Okay, so these are pictures of microbes underneath an electron microscope here. They're shaped like a rod, so they are bacillus bacteria. And we have done studies where we find that these two types of bacteria here can increase the production of short-chain fatty acids, which have direct effects on the immune system. So when you have these types of bacteria, you have less oxidative DNA damage, you have tumor suppression, and you have basically a healthier environment than if you did not have these microbes. So this is just the immune system. We also see that certain gut microbes can contribute to the anti-inflammatory program. So if anyone has heard of these inflammatory bowel associated diseases like IBD, Crohn's disease, colitis, even C. diff, Clostridium difficile, is associated with a lot of inflammation, and this is antibiotic induced. So this is usually an infection that will arise after an antibiotic treatment that allows the bug to overgrow. But my point here is that certain gut microbes can modulate this anti-inflammatory program and can lead to disease. And we find that Bacteroides fragilis, this is a type of bacteria, plays a major role in determining outcomes of inflammatory molecules in the gut. Surprisingly, and this has just shocked the scientific community, and I think it's still kind of a little controversial, but I wanted to share this with you guys because I just think this is so cool, that microbes that live in your gut not only can predispose you to certain inflammatory related diseases, but they also seem to play a role in mental states, behavioral states, and even we're finding links with certain spectrums of autism or certain disorders associated with autism, in the sense that there are different bugs in a healthy person's gut as compared to someone who's suffering from a particular mental illness or even autism. And we also see this correlation in these stress states with an increase in 4-ethyl phenyl sulfate, which is a product that can be lowered by adding Bacteroides fragilis to the diet. So this brings in this whole concept of probiotics and how powerful potentially this may be in the future for treating human disease, even heart disease. So who would have thought that the microbes, right, the normal microbes that live within the human body could be predisposing you to developing heart attack, stroke? And this is all has to do with, we found links with this bacterium called Prevotella. So if any of you have heard of this notion that your oral health is linked to your heart health, well, Prevotella is a bacterium that causes a certain form of ulcerative gingivitis, and it's found in the oral cavity and can spread to the bloodstream and can contribute to higher levels of this TMA and TMAO, trimethylamine N oxide, and trimethylamines, which can perpetuate an atherosclerotic condition. So, take-home message from this, certain microbes, normal microbes, can actually perpetuate heart disease. They also can perpetuate diabetes or may even be linked to a causative agent in the development of diabetes and also obesity. You can take a look at someone who has the condition diabetes or is obese. You can look at the microbes that live in their guts and compare them to people who are leaner, healthier, weight-wise, and you will find differences in butyrate levels and fatty acid oxidation, all related to the types of microbes that are present in the gut. And if you start to modulate these types of microbes, then you also see concomitant changes in disease phenotypes. So basically my point here is that I would like all of us to realize that the 90% of microbes that make up individually who you are playing a critical role in your health status. And our lifestyle, the choices that we make in our life, can determine how those microbial populations fluctuate. So we have someone here at a restaurant saying, I'll have the garden salad, please. But his microbes that are living in his gut really want cheeseburger and fries because he's been living off of a diet potentially more dependent on bad foods. And so you can actually select for these microbes. You can change the microbial populations. And I wanted to share this simulator here that actually shows examples of how the normal microbes that live in the gut can change over time and how this can determine your health status. So all of these antibiotics, fever in response to an infection, being sick, so being infected with a microbe that causes disease. This is referred to as a pathogen. What you are eating and what you're putting into your body on a daily basis. What do you eat when you wake up in the morning? What do you have for lunch or dinner? All of these things, usually people are consistent with what they eat, and changing your diet or maintaining a certain diet can influence what's growing in your gut. Also, developmental hormonal changes cause fluctuations in microbial populations as well. So I'm just going to show you guys a couple examples of this. So this is a simulated, let's say a simulated gut environment, right? So you have all these populations of microbes. I said there's potentially over 10,000 species that are living in your guts. And you have these bacillus, bacteria, rod-shaped. You have chains of bacteria called strep. So if anyone's heard of strep throat, strep bacteria exist as chains, okay? Or you could have these little squiggly guys. These are spiral or spirillum. And the little spherical shaped ones, coccus, okay? But all of these are unique, different strains of bacteria. So let's say you become infected with strep throat and you are prescribed an antibiotic. What happens to your microbial populations?
Antibiotics are deadly, but only to some bacteria. Certain bacteria are affected immediately, and oftentimes shifts in these bacterial populations affect other community members. The effects of antibiotics can be dramatic and long-lasting.
So you get the idea here that antibiotics are very powerful. They will kill off certain targeted strains and populations of bacteria, and this is something that can have a lasting effect. Now, I said before that certain strains of bacteria are responsible for producing or metabolizing disease-causing compounds. So when you take antibiotics, you're really changing more of a long-lasting effect of your health potentially in the future. What happens if you change your diet?
The food we eat is also food for the microbes that live in our digestive tract. Depending on the nutrients we eat, different types of microbes will increase or decrease in number. Sometimes new microbes enter the body along with our food.
So you see there's an increase here in the population of this type of bacteria, right? So if you completely change your diet from eating meat all the time to vegan, you're now asking your gut to metabolize certain nutrients and certain compounds that it really hasn't been seeing a lot of. And so you can actually select for which microbes are preferentially growing in your gut based on what you're putting into your body. So this is just an example here of how, again, lifestyle choices can affect or can modify these microbial populations. And just to the point that I mentioned before, that microbes are everywhere. For example, in your kitchen, right? So here's an example of on one floor in an apartment building, Peter scrubs away the scent of Len's dog, and then he scrubs a little more. He manages to destroy many of the microbes brought in by his neighbors at his housewarming party the night before, but wasn't necessary. Some bacteria, such as the one that causes fatal outbreaks of Legionnaire's disease, deserve to die, but others killed in our war against microbes might be the ones that are keeping us healthy. So not all of them are bad. On a different floor here, you see, remember I mentioned that microbes are everywhere, right? So on the second floor of an apartment building, Gary, an environmentally conscious vegan, keeps half of the floor cool, preferring to save energy. Larry, a glutton, turns up the heat. Neither realizes their differences reach far beyond lifestyle and stretch to the microbes that surround them. Hot and dry rooms have been found to harbor lots of Staphylococcus epidermis, which is a common skin microbe, while the cooler rooms contain more Ralstonia piquetti, which is a bacteria that's common in water, soil, and on plants. So in other words, Gary's microbes are tree huggers too. So I'm showing you guys this to illustrate the fact that microbes are everywhere. They're all around us. They're within our bodies, on the surface of our skin, and we can select for microbes that are not only in our environment, but also microbes that are maintaining our health. Now, we've already noted a lot of microbiota human disease links. So I shared with you guys autism, also even depression and anxiety. We find certain strains of bacteria in depressed people's guts, or people who are suffering from certain anxiety disorders. Also asthma, heart disease, peripheral vascular disease, colon cancer is a huge one, obesity, and a lot of the inflammatory bowel or inflammatory associated diseases. And this is just so awesome. So I don't know if any of you guys have heard about this. I know there was a special on NPR and a special documentary on CBS, a 60 Minutes special on this. But recently we have seen that in medicine, treating patients who are suffering from what's called C. difficile infections. So C. difficile, this stands for Clostridium difficile. This is actually a pathogenic, a disease-causing bacteria that is actually normally found in a small amount in the gut, but when patients are treated with antibiotics for other diseases, this C. diff will overgrow. And when it overgrows because of dysbiosis, you end up with Clostridium difficile infection. These patients have severe diarrhea, abdominal pain, nausea, and fever. It can actually kill you. And what they're doing now is they're taking the feces from a healthy individual and they're transplanting it into the gut of people who are suffering from C. diff, and they're finding that it cures them. So what does this say? Again, this is illustrating my whole theme here, that your gut health, changing the microbes that are growing in your gut, can determine your health status. So in this case, good microbes transplanted in the gut can actually improve the immune system and can battle C. diff infections and can cure people of this infection. And remember, everyone's different, right? So we are all unique with the types of microbes that we have. Again, each bar is one single human sample. And check it out, everyone is different. So this is just so cool. So congratulations to all of you for being able to learn about this stuff at this time. I mean, I just think it's awesome, a very awesome time to be in the field of medicine that in the future, you know, we've heard a lot about DNA, getting your DNA sequenced, and that's also a wave of the future. But I do believe that in the future we will not only have our genome sequenced, but we will also have our microbial profile determined, and we will be able to treat an individual based on not only what their unique human genome displays, but also what the microbial genome displays as well. And I wanted to share with you guys an ongoing project. This is actually at a university in Colorado at UC, and you can participate in this study if you want. You can go to this website and you pay $99 and they'll actually send you a kit. You can send a sample of fecal matter, swab your mouth and your skin. And you also can take samples from your pet. You can send this to the sequencing facility at UC, and they'll do the sequencing and analysis, and they will tell you what is your unique selfie microbial profile and how do you compare it to everyone else in the US. So this is a really cool project that's going on right now. So that was kind of my little spiel about normal microbiota, and just to make all of you aware of how prevalent and how present microbes are in our lives. And we don't even realize it. We don't realize it because microbes are too small to be seen with the unaided eye. They are microscopic, okay? We can grow them in the lab, and we would use something called an agar plate, which you guys are going to be working with this week or next week, depending upon when you guys start it. But you're going to be making agar plates, and they're basically these plastic petri dishes containing agar. That's the background substance here. Agar is unique in the sense that it has this chemical property whereby if you heat it up to boiling point, it will be liquid, and at room temperature it turns solid. So we can heat this stuff up, we can melt it, and then we can pour it into a petri dish, which is what you're going to be doing. You let it sit and it will solidify. So once it's solidified, you start off with this agar that is sterile. There is nothing growing in it. There are no microbes there. When you pour it into the plate, anything from the air or anything that you introduce from whatever experiment you're setting up will introduce and contaminate that agar plate. Okay, so in this case, this agar plate has been contaminated and has been inoculated with different strains of bacteria. All of these dots that you're seeing here are what we call bacterial colonies. So remember, I pointed out that CFU before, and I said that standard for colony forming unit. Each one of these circles is a colony forming unit, and each colony is defined as a colony such that in this one little group of cells, all of the cells that are growing in this little dot have been derived from one cell. So when we introduce bacteria to this plate, one cell fell here, one cell fell here, one here, and so on. And after we let these bacteria incubate, they grew, that one cell divided to produce two, two to produce four, etc. And then in the end, when you have about 1 million cells in this little spot, you can see them growing on the plate. But this one colony is not representative of a single cell. If we were to take a sample of this and put it on a slide and look at it under the microscope, we could then see the single cell, which would look something like this. This is a single bacillus bacterial cell. This is magnified 300 times. It's pink because it's stained. You guys are going to be doing a lot of staining this semester. You have to stain the cells to see them. You can't see them if they're translucent or transparent. You have to stain the cell a particular color. So we use microbes to produce and synthesize all of the antibiotics that we've ever been prescribed. They regulate all of the biogeochemical cycles around us. So this means the carbon cycle, all of the processes that regulate ecosystems, the sulfur, nitrogen in the environment, in the soil, how nitrogen is fixed from nitrogen gas into the soil environment. And for anyone out there who likes to enjoy a beer or a glass of wine or cheese, all of those are synthesized by certain microbes. So Saccharomyces will ferment starch and will produce carbon dioxide and ethanol. So you can make wine and beer that way. You also need lactic acid bacteria to make cheese. Okay, so microbes are contributing to our environment in ways that we don't even realize. And again, we can't see them, right, with the unaided eye. We need to visualize them under the scope. Now, the downside to microbes are that some can cause disease, right? So this is a huge part of, for those of you who will be actually practicing medicine, working as nurses, working in the medical field, you're going to see a lot of cases of infectious disease. So an infectious disease is basically a disease caused by a pathogen, and a pathogen is a microbe that can make a person sick, okay? But remember, the majority of microbes are actually good for us. So some can cause infectious disease, but some also produce, like I just said, lactic acid, ethanol, and as major producers, they produce oxygen. If they are photosynthetic, they're functioning as autotrophs in the environment, but also acetone, vinegar, also bread, and collectively, we can use microbes to engineer them in a specific way to produce whatever product we want. So we can collectively take a population of microbes and engineer them, clone in whatever gene that we want to produce whatever protein product we want. In this case, this is showing you insulin. We do use yeast and bacteria to make insulin that we then will use to treat diabetic patients. We can use microbes to stonewash, so this is a form of a fungus, bacteria for cotton. Certain dyes can be produced by bacteria. Also, there's a lot of research going into using bacteria to produce new forms of plastics or alternative forms of plastics, since some research is coming out saying that plastic, the traditional plastic that's used, leaks out BPA or bisphenol A, which can be toxic. So they even suggest pregnant women do not drink out of water bottles or plastic water bottles because of the exposure to that. Now, we've come a long way. So just imagine in the past, hundreds of years ago, when we were blind to the fact that microbes even existed. So the early microbiologists that claimed that we had these bugs crawling all over our skin, or these germs that lived within our guts, I mean, those people were stigmatized and ostracized as being lunatics. And now we're completely aware that microbes are very prevalent. They're present in our lives, in our everyday lives. So having and gaining knowledge of microorganisms has allowed us to not only treat people with infectious disease, but be able to prevent the spread of infectious disease and also prevent food spoilage. So we know that when you leave bread out for too long, there's some fuzzy stuff that will grow on it. That is a specific type of a microbe, usually a fungus that will grow. And we are completely aware of how that happens, why it happens, and what the types of microbes are that will spoil food. Now, we also, through the knowledge of how pathogens can cause disease, we have created the advent of aseptic techniques. So this is so critical when you guys are working in the medical field to know how to prevent contamination between rooms, between patient rooms, how to work with sterile medical devices and instruments, and how to prevent yourself from getting sick, right? So people are in hospitals because they're sick. It's a perfect breeding ground for certain infectious diseases. Now, just a little bit of history here. So very important that you guys are all aware of how microbes are named and classified. So I would remember Carl Linnaeus established this basic system of scientific nomenclature. And the biggest thing to remember is that whether we're talking about a human, a cat, a dog, a horse, or a bacterium, they all follow along this standard system of scientific nomenclature, including a genus and a species or a specific epithet. So Homo sapiens for human beings. Bacillus anthracis is the causative agent of anthrax. So even bacteria have their own naming system of genus and species. And all of these names are always italicized. The genus is always capitalized, and the specific epithet or the species is lowercased. They're used worldwide, and they may be descriptive or honor a scientist. So Theodore Escherichia is the scientist that discovered E. coli or Escherichia coli. So it was named after the scientist. Coli describes the colon. You also may see, I mentioned Bacillus anthracis. Sometimes bacteria contain a descriptive word that will give you information about the shape. So bacillus refers to a rod. If you've heard of strep throat, streptococcus, or staphylococcus, if you are a strep bacteria, you exist as a chain. If you are a staph bacteria, you exist as a cluster. And coccus is referring to spherical shape. So here's some examples. I just said Escherichia coli, honoring the scientist, colon, in the name coli for the habitat. Staph aureus describes the clustered shape of the coccus, and also aureus is the color, the gold color of the colonies. Now, throughout the semester, we're going to be looking through and learning about all of the different types of microbes that exist in the world. So bacteria make up a huge percentage of the microbes that are in your surrounding environment. Also archaea, so these are old, archaic for old ones. These are similar to bacteria in the sense that they're prokaryotes, but they live in extreme environments. So you would find an archaic cell living in a hot spring or in a salt lake where typical bacteria would not be able to thrive. Also fungi, there are multicellular and unicellular types of a fungus. Protozoa is another group. These are eukaryotes and they're larger than these two groups of microbes. Algae, which most algae do not cause disease in humans. There are some strains of algae that can release neurotoxins in the case of, if you've heard of red tide, that can cause food poisoning. But for the majority of these cells, they are non-pathogenic. Viruses. This is one of my favorite group of microbes. They are acellular. They need a host to proliferate or to replicate, and viruses are ubiquitous along with bacteria. Some microbiologists would claim that there are more viruses in the world than bacteria. And then also multicellular animal parasites. So you might be wondering, well, why are we going to be learning about animal parasites? These are worms or helminths. They're parasitic forms of worms that have these microscopic life cycles. So they can cycle from an egg to a larva to an adult. So we will be learning about the different types of helminths as well. Now, out of all of these types of life forms, they fall into one of the three following domains of life. The only exception here is viruses. Viruses are considered acellular, so they actually don't fall into any one of these three domains. But the rest of them, bacteria here, right, the one on the top of the list is in the bacterial domain. The archaea are in the archaic domain. And the rest of these guys, fungi, protozoa, algae, and helminths are all in the eukarya domain along with us. So these are all the eukaryotic cells. Now I'm going to go through and give some specific focus points of each type of microbe that you should be familiar with, at least for week 1. Bacteria, they are prokaryotic, meaning that they do not have a nucleus, they're small, they don't have organelles, and major point, all bacteria have peptidoglycan. Peptidoglycan is a disaccharide. and it gives rigidity and structure to a cell wall. Basically consists of this scaffolding of these NAG and NAMs, which we'll learn about later. These are basically certain forms of acids that intercalate with one another and provide the basis for the cell wall. Bacteria divide via binary fission, so this is basically a fancy word for just dividing right down the middle. They will replicate their DNA and divide down the middle. And there are many different diverse types of bacteria that can use organic, inorganic nutrients as a source of energy, and also some are even photosynthetic. Archaic organisms, they are prokaryotes as well, but remember they lack peptidoglycan, and I mentioned before you always find them living in extreme environments. So some are methane producers, they can withstand high salinity, high temperature, high pressure. So remember, old ones, they live in extreme environments. And then we also have fungi. There are multicellular and unicellular forms. So yeast, the Saccharomyces cerevisiae that's used to brew beer, is a single-celled unicellular fungus. A lot of these you find growing in soil environments. And actually the first antibiotic that was produced, penicillin, was produced by a fungus. So Penicillium produces penicillin, and this is a single-celled fungus. When you see it growing on, let's say you leave bread out for too long and you see the mass of the furry stuff, that's referred to as a mycelia. But if you were to take a piece of that off of the bread and look at it under the microscope, you'd see these smaller units composed of filaments, and these are referred to as hyphae. Also, protozoa. So these guys are eukaryotes. They have a nucleus, they have organelles, and these guys are really cool to learn about because they have these complex life cycles. So if anyone has heard of toxoplasmosis, toxoplasmosis is caused by Toxoplasma gondii. It's a parasite that has this very unique life cycle. And if any of you have heard of the crazy cat lady phrase, that people who own a lot of cats are not the most mentally sane people. Well, there's actually some scientific evidence behind that. There's a certain type of protozoan, that Toxoplasma gondii, that cycles from rodents, so a mouse to a cat to humans. And it's very interesting that when a mouse is infected, it changes the brain chemistry of the mouse and makes the mouse attracted to its predator. So it will actually seek out a cat, the cat will eat the mouse, the cat becomes infected with the parasite, and the cat will shed the parasite in its feces. So that's why they say pregnant women should not handle cat litter. If a pregnant woman contracts that parasite, it actually can cause stillbirth. But generally, if people are infected with that parasite, we find that there's higher rates of mental illness and suicide in those types of infections. So the crazy cat lady, there's some scientific evidence or basis behind that. But my point here is that protozoa, they're very interesting because they usually exhibit these complicated or complex life cycles. Also, algae. Algae are eukaryotic. They utilize photosynthesis for energy. And what I would remember here is that they are able to, via photosynthesis, produce oxygen and sugar and organic compounds. And there's a picture here of green algae, and you see the pigment green from chlorophyll, and there's some really cool different types of algae, diatoms, that have these diverse shapes that look like little pinwheels or spiral shapes. Some of these can actually cause, like I said, can lead to food poisoning if you consume the toxin, usually associated with shellfish, but the majority of algae are non-pathogenic. We need them to produce oxygen in the environment. And then we have viruses, which I think this is my favorite topic. Viruses are considered acellular. Again, they're not part of any one single domain of life. And basically, the way that a virus perpetuates itself is it needs a host cell. So if a virus is hanging out on a doorknob or a surface, it cannot replicate itself. You cannot take a virus and inoculate it into an agar plate like I showed you guys before on the surface of an agar plate. A virus will not grow. You need to actually induce replication by introducing this virus into a host cell. So you need to infect an animal with a virus to see more copies being made, or you need to infect cells, a cell line. And viruses are, I consider them little molecular machines. They have either DNA or RNA. So they have a form of a nucleic acid. They have a genome, which is either deoxyribonucleic acid, DNA, or ribonucleic acid. They also have a protein coat. They may have an envelope. So if they don't, they're called naked or non-enveloped. And again, they need a host cell to replicate. So they are what we call obligate intracellular parasites. And there are some that cause some nasty diseases like Ebola, which now is still raging across West Africa. And a lot of viruses, we don't have the best treatments or cures for diseases associated with them. Also, we have the helminths, the worms. So you can see these guys with the unaided eye, right? So here's a picture of a heart infected with a heartworm helminth parasite. If it is a worm causing disease, it's referred to as a helminth, and they do have microscopic life cycles. So they will cycle from an egg to a larva to an adult, which is this form of the worm that you're seeing here. Now, I want to talk very briefly about early days of microbiology in a time where, again, people were so blind to this whole concept of smaller units of life, right? So people didn't believe anything unless they saw it, and you can't really blame them. This is why the microscope was such a groundbreaking invention, and the use of the microscope, along with all of its associated discoveries, really changed our whole perspective of the world around us. So kind of the beginning of this in the stages of infancy, 1665, Robert Hooke reported that living things were composed of little boxes or cells. So he actually took a piece of cork, which is dead plant material. He looked at it under the scope and he saw these smaller units of life. These were dead cells. But it wasn't until some of the later experiments, Rudolf Virchow, 1858, found that cells arise from pre-existing cells. He actually had his grad student watch cells divide under a microscope slowly, painstakingly, over 20 hours, watching one cell replicate its DNA and divide to make a whole new cell. But this provided the basis of cell theory, that all living things are composed of cells and come from pre-existing cells. And Anton von Leeuwenhoek kind of followed up with Robert Hooke's discovery of these smaller units of life. He looked at live microorganisms, and he used this makeshift early version of a microscope, which obviously now we have these amazing modified advances in the microscope that allow us to magnify certain objects up to 1 million or more times. Now, there was this early debate over spontaneous generation. So related to this concept of cell theory, this was not agreed upon right away. So there was a time in our history where we really didn't know where life came from. So there was a hypothesis that living organisms arose from non-living matter. There was this vital force that life just spontaneously appeared from nothing, out of thin air. And this was soon disproved by biogenesis. So biogenesis and experiments that were conducted supporting this said that you need a cell, a pre-existing cell, to produce a new cell. So living organisms have to arise from pre-existing life. They can't just be derived from nothing. And Louis Pasteur conducted an experiment that I want you guys to remember, this S-shaped flask experiment. essentially what he did was he invented this type of a flask. And this was the key for this experiment, is that it had this swan neck. So there were a bunch of early microbiologists that would conduct experiments where they would take jars, fill the jars with meat, and they would let the jar sit out, and they would see if the jar would become contaminated with larvae or mold or bacteria over time. And they did certain experiments where they would actually cover the jars with nets, and they would see what's the difference between a covered jar versus an uncovered jar. So whatever is growing on the meat must be coming from the environment. Because in the case where they saw the covered jars, they saw that nothing would grow on that meat. Now, this was a similar experiment, but what he did was, with regards to bacterial contamination, he created this S-shaped flask. And the key here was that it was open at the end, so air could get in, but whatever air could get in was trapped here at the bottom of this swan neck. And so what he did was he boiled this nutrient broth to sterilize it. This is basically the whole concept of pasteurization. And over time, nothing would grow. But in the case where he had this swan neck flask where he would boil it and then chopped off this piece where air could get in, it would be contaminated. So the point was that whatever was growing in the flask or contaminating the media was derived from the external environment. It was derived from something floating in the air, which we know now that there are microbes that are attached to dander, hair, dust that's floating in the air. And that's why it's so critical when you guys are doing these experiments that you realize that fact. So when you're making these agar plates and you're doing these experiments in your home, please, when you're pouring the agar plates or you're opening them and you're introducing microbes to the plates, you need to realize that if you keep that plate open, there's dust and dander and microbes that are going to contaminate that surface if you leave it open long enough. If you have hair that's hanging over the plate or you sneeze into the plate, you're going to have stuff growing there that shouldn't be there. So please be aware. Please try and practice sterile technique and reduce contamination. Now, I wanted to share with you guys this golden age of microbiology. So for week 1 here, it's really important that I cover some of the most important contributions made by early microbiologists. And a lot of these are derived from this specific span of time, from 1857 to 1914. It really started with Louis Pasteur's work. And once we discovered that there were these smaller units of life, we were then able to figure out how maybe some of these smaller units of life were causing disease. How could we protect or prevent people from contracting certain diseases? And this whole concept of immunity arose out of this era. So Louis Pasteur discovered that microbes can spoil food. So this is through the fermentation process. Fermentation is the conversion of sugar to alcohol. Anyone, again, who enjoys a glass of beer, this is the result of the fermentative process. So this is from yeast fermenting certain forms of starch or sugar. And also food spoilage, like I said, growth on the surface of certain food types in the right environment can spoil certain foodstuffs or even beverages. And what can happen here, so I myself I am a beer aficionado. I like to brew my own beer. And what can happen here is that when you brew your own beer, you want to make sure that you have a pure, sterile culture of yeast that you're introducing into the beer when you're brewing it, and that it does not get contaminated with certain strains of bacteria. If it does, the bacteria can take that alcohol and convert it to acetic acid or vinegar, and it can spoil the wine or the beer. So there are certain microbes that will carry out that process. Now remember, pasteurization, this was devised by Louis Pasteur himself. He found that just heating up a specific substance for a certain period of time would reduce the amount of microbes present in that substance. And we're going to be talking all about different forms of antimicrobial control later in the semester. But more discoveries during this era. The whole germ theory of disease. So Semmelweis advocated hand washing. Oh, this is huge. This is a very common mode of transmission through direct contact of whatever microbes you have on your hands, the transfer of those microbes from your hands to another surface. So you have disease-causing microbes on your hands, you touch a doorknob, that can be transferred to another individual, and then that can lead to disease. So we now know that this is a huge preventative measure. This was not realized until the 1840s. 1835, 1865, we had early microbiologists looking at different smaller forms of life that they thought were implicated in diseases. And 1860s, applying Pasteur's work, we started using aseptic technique, and Joseph Lister really is the guy that advocated for the use of an antiseptic during surgeries. And this early chemical disinfectant was actually carboxylic acid. It was super caustic and strong. Now we don't use that. We use iodine, alcohol, other forms of antiseptics, but Listerine was named after the early microbiologist Joseph Lister. I would also like you to remember the early microbiologist Robert Koch. This was in 1876. He devised the set of Koch's postulates. So these postulates allowed early microbiologists to determine if they suspected that there was a specific microbe that caused a disease, they could follow along these postulates to make the claim that yes, Mycobacterium tuberculosis, for example, causes tuberculosis. How do we know that it causes that disease? Well, he devised these early postulates that I'm going to be going over later in the semester, and as we'll see, there are exceptions to these. Where did the idea of a vaccine come from? 1796, Edward Jenner. He actually conducted this taboo experiment. He inoculated this young boy, whom he got permission from the mother of the young boy to do so, with cowpox. So he took a sample from cowpox, which is very similar to a virus that causes smallpox in humans. He inoculated the boy with the version of cowpox, and the idea here was that cowpox was an attenuated form of smallpox. And this is the basis for how vaccines work. So cowpox is an attenuated version of smallpox, meaning it's weakened. So it's very similar to smallpox, but it's weak enough that it allows the native human immune system to synthesize antibodies in time so that once they are exposed to the full severe form of smallpox, that they have the appropriate antibodies to combat that infection. And we're able to devise our own vaccines in the lab by figuring out how can we weaken a certain pathogen or mutate it enough so that the native immune system will still be able to produce antibodies, but it won't cause specific infection. And this is how we produce a lot of the vaccines that we use today. Even the flu vaccine, some vaccines have live virus or live pathogen. Others may just have pieces of the pathogen that will induce the production of antibodies. Out of this whole wave of microbiology early wave of microbiology came the idea of using antibiotics or chemicals to kill pathogens that are causing disease. Now, what I want to stress here is that antibiotics are actually naturally produced by bacteria and fungi. So if you've ever been prescribed Z-Pak or penicillin or erythromycin or tetracycline, all of those are derivatives of compounds that are naturally produced by microbes. Why does this make sense? Because microbes want to produce those chemicals in their native environment to compete against other microbes for space and for food. So it's a selective advantage. A bunch of other early studies here. Paul Ehrlich speculated about a magic bullet that could destroy a pathogen without harming the host. So this is the whole idea of an antibiotic, right? It's a perfect idea. Take an antibiotic, it'll just target the bacteria that's making you sick, but it won't target your human cells. Well, now this kind of whole idea of antibiotics being magic bullets has really been turned upside down. Hopefully you guys are thinking that way from the first half of this lecture when I said the normal microbes that live in the gut actually maintain our health, and antibiotics have more long-lasting, drastic effects than we originally thought. First antibiotic, please remember, it's penicillin. It was discovered by a serendipitous accident here. So this was kind of a fortunate accident. We had an agar plate that had bacteria growing on it. Alexander Fleming had inoculated this agar plate with different staphylococcal bacteria. So this is Staph aureus bacterial colonies, and serendipitously it got contaminated. So remember I said that there's stuff flying in the air, right? Attached to dust and dander, that microbes are everywhere. The plate got contaminated with a fungus, and what he found was after a day of incubation, he looked at the results and he looked at what was growing, and he only wanted to see bacteria growing, but he saw there was a clearing of bacteria where this plate had been contaminated with a strain of fungus. Now, what we know is that this fungus produces an antibiotic called penicillin that specifically targets staph bacteria. Now, go figure, we have a lot of antibiotic resistance. The first notorious antibiotic resistant strain was MRSA, right? Methicillin resistant Staph aureus. Why? Well, penicillin was the first antibiotic. It's been used for over 50, 60, 70 years. It's been used to treat a lot of skin infections, and now because of overuse, we're seeing a lot of resistance. Out of the early studies of microbiology and discoveries, now we have all of these unique fields dedicated to just studying bacteria, just studying fungi, or viruses, or parasites. Now, throughout the course of the semester, we're looking at all of these, right? So we're not just zooming in on bacteria that cause disease. We're going to look at all different types of microbes that are beneficial for humans and also that are detrimental and can indeed make you sick. We learned in the early days this whole concept that our immune system, depending upon the individual, is different, right? So your immune system is unique as compared to the next person in the room. So immunity, it's a complicated topic of study here that I will go over some basics of antibodies that are produced in the body as a result of or in response to infection. But the main thing really to take home from this is that depending upon your unique immune system will determine different types of infections that you may come down with. And there's different types of immunity. There's passive, active, acquired immunity. There's immunity that can be developed over time that's determined by the normal microbes that live in your gut and also by vaccines that have been administered to you over the course of your lifetime. A lot of cool stuff related to recombinant DNA technology with the knowledge and the awareness that microbes existed. We've now, you fast forward 100 to 150 years, and we're now at a time in our history where we can manipulate microbes genetically to produce all different types of compounds. We can take genes from different species. We can take genes from humans or genes from other animals and clone them into bacteria, and we can have the bacteria produce a human form of a protein or a human form of a compound. And then we can purify and isolate that compound for our use. We can trick the bacteria to pass on certain traits, these certain traits that will allow for resistance or a lack of resistance. And this all lied in the ability of us to be able to determine what the DNA sequence of every organism was. What is the concept of a gene? A gene is made up of DNA. That DNA is basically life code that tells the cell to do everything. Okay, carry out every single process that it is able to carry out. And by manipulating DNA, we're manipulating life and how that organism is acting. So very powerful technology based on the discovery of molecular biology, which says that DNA codes for RNA, which then codes for protein. And so these were discoveries in the 40s, and 50s and 60s. In the 50s, we discovered the structure of DNA, and we really have come a long way. If you guys have heard of GMOs, right, genetically modified organisms, we're actually creating novel species that we've never seen before, utilizing bacterial DNA messages to do, again, anything that we want. So produce any antibiotic or compound that we could exploit for its use. We will talk a little bit about environmental microbiology and how important fungi and bacteria are for recycling elements in the environment: carbon, nutrients, nitrogen, sulfur, phosphorus. These are all used by plants and animals, and microbial processes are key for recycling these. We have bacteria that can actually clean up oil spills. They can clean up toxins in the environment, and we can genetically engineer them to carry out specific functions. So certain strains of bacteria can withstand these extreme environments, like Deinococcus radiodurans is literally found living in nuclear reactors. There are types of bacteria that will eat hydrocarbons found in petroleum for lunch. They metabolize it like it's a nutrient, and they can take that toxic compound and convert it into something that is non-toxic, that will not lead to detrimental or long-lasting negative effects on the environment. And this would be anything from an oil spill to mercury that builds up in the food chain. GMOs, I mentioned this before. A great example of this is certain plants, insect-resistant tomato plants. This is a scheme on the right here. There's a strain of bacteria called Bacillus thuringiensis that produces a specific toxin that is toxic to insects, and it's not toxic to humans. So what we've done is we've created plants that are GMO by cutting out the gene found in this bacterial cell that will code for that specific toxin. And we can take that gene and we can use, there's different methods of doing this, but you can introduce that genetic material into plant cells. You can select for embryos that contain that specific toxic gene, and those plant cells will perpetuate and grow into a mature plant containing the bacterial gene, and they will produce the bacterial gene product so that when an insect is a pest to that plant, it will kill the insect, but it is non-toxic to humans. So this is an example of really the power of how far that we've come by learning about the DNA of bacteria and exploiting certain functional attributes for our benefit. Microbes have been used for centuries to produce many different foods and chemicals. So I already brought up beer and wine. Cheese is another one. Along with this, recombinant DNA technology. So this is related to the GMO idea that I just said, but we've also used recombinant DNA technology to produce all of the vaccines and certain enzymes that, for example, in gene therapy models or GMO models of plants to protect crops from pests or from drought resistance. All of this has been made possible by studying the genetic material of microbes. And microbes are so easy to manipulate. So no one cares if you kill millions of bacteria, but it's not that easy to do with rodents, small mammals, or humans. So a lot easier to do those types of experiments with small microscopic life forms. Now, the majority of my lectures so far, I've tried to convince you guys that microbes are fantastic. They regulate our health, and we use them, we can exploit them for all of these other functions and commercial functions in our environment, industrial applications. But there are some downsides, right? So they can make us sick. They can cause infectious disease. And now we're seeing with the advent of antibiotics, we see some resistant forms, and also they can produce biofilms. So what is a biofilm? This is basically a highly resistant form of bacteria that does not respond to the typical antibiotic or antimicrobial treatment. So a biofilm, if you run your tongue over your teeth and if it's slimy, you probably have a biofilm of bacteria on your teeth. That is why teeth cleanings are so important. That's why people get cavities. That's why gingivitis and oral disease exists, because bacteria will adhere to the surface of your teeth, and it forms a very, very sticky, highly resistant layer. They also can form on catheters, medical implants, medical devices, rocks, pipes. And there's this whole process that we've studied over the years. We know how it evolves. So it kind of starts off as these sessile, free-floating cells, which then adhere to a surface. And over time, there's cell-to-cell adhesion, not only cell to the object, the inanimate object adhesion, but cell-to-cell within the microbial population. And some of these cells can actually disperse, they can break off, and they can go and attach to new places in the surrounding film. Very notorious bacteria here, staphylococcal biofilms on catheters, resulting in UTIs. Now, when a microbe causes disease in a human, it's called a pathogen. And I wanted to share with you guys just for week one, and we're going to get into a lot more details about different types of emerging infectious diseases, or all of the different types of infectious diseases. But I wanted to just mention some that seem to be increasing in incidence. And they're a little scary because we really don't have appropriate treatments or cures for them. So everyone freaked out, and there was a lot of hand waving when the epidemic of the swine flu arose a few years ago. So you heard on the news, watch out for the swine flu. It's a new strain. Well, swine flu is actually H1N1. The H's and the N's are referring to these spikes on the surface of the virus. So H is hemagglutinin, N is neuraminidase. And these are specific components of this pathogen that play a role in how they will attach to your cells and infect your cells. Now, H1N1 can infect cells of the respiratory system. So this is swine flu, and the reason why people were pretty much concerned with it was because it was novel in the sense that it had swine genetic material, which we hadn't seen before. But H1N1 was actually circulating in 1918, and that's why some people who were older of the geriatric populations were actually potentially protected against it, whereas young were more at risk. But H5N1 is the bird flu. This is the one that you want to worry about because it has a 50% mortality rate, and it would cause a worldwide pandemic if it gained the ability to jump from human to human. Because that has not happened, we haven't seen a pandemic, but this is a disaster waiting to happen. So I just said it's a 50% mortality rate. The average flu does not kill half of the people that are infected, right? It's a much lower rate. So bird flu is the one that you want to be worried about. MRSA, methicillin-resistant Staph aureus. There's a ton of this running rampant in hospital environments. Why do we think this is the case? Well, I said to you that penicillin was the first antibiotic that was discovered. And back in those days when we discovered that we had a compound that could be used to treat any infection, doctors were overprescribing it. And there's been this great increase in abuse of antibiotics, overuse, and just an abundance of antibiotics in our environment. When you go to the grocery store and you buy chicken or you buy beef, the meat that you're actually purchasing is coming from animals that have probably been treated with antibiotics. The farmers are giving their animals antibiotics so that they don't get sick, but we are consuming those compounds on a daily basis. And that's just perpetuating this resistance epidemic. And I do believe this is going to be a huge problem. There are 23,000 people who die every year of antibiotic resistant strains now. It's estimated and projected by 2050 that millions will die every year. So this whole resistance epidemic is something to be concerned with. West Nile encephalitis. We don't really see a lot of cases of this, but just in general, diseases that can be spread via insects are on the rise. And the way that the incidence fluctuates with regards to these diseases is dependent upon the insect populations. So when you see climate change and you see an increase in insects in that environment, you will also see a concomitant increase in the numbers of these types of infections. Now, West Nile is scary because it can lead to fatal encephalitis or swelling of the brain and CNS infection. We saw it in 1999 in New York City for the first time, and we've had some cases even here in Connecticut. We don't have a cure for it. This is another very interesting infectious disease that I think is one of my favorite to talk about because it's rather anomalous. We don't know a lot about it, and it's caused by this entity which I haven't brought up to you guys yet. It's not considered a microbe itself, but it's called a prion. A prion is actually an infectious protein. So this is really an outlier among all of the microbes that I mentioned to you guys, because everything that I mentioned contains cells, right, except for a virus. A virus is acellular. But a prion is even smaller than a virus. It's just a single protein. And surprisingly, we actually all have a normal prion protein that is circulating in our bodies. We find it in our bloodstream, certain organs, in our brain. And when we consume a bad version of this protein by eating bad meat, I don't know how many of you have heard of mad cow disease, but there was an epidemic of this in London in the 80s, and they ended up having to slaughter hundreds of thousands of cows that were infected with this. But if you consume meat, let's say you go to McDonald's, you get one of their double cheeseburgers, you may be consuming spinal material from a cow that is already infected with mad cow disease. If you consume that bad version of the protein, the prion protein will convert all of your good prion protein to the bad form. And this will basically eat away at your brain. So when it eats away at your brain, it causes Creutzfeldt-Jakob's disease. You have 3 months to live. It's very rare, but there's no cure. It's very fast acting. And the pathology is similar to Alzheimer's, where the brain basically contains these fibers that will build up. And the only way to diagnose this is via an autopsy. You'll actually see the brain will look like a sponge. And that's why it's called spongiform, because it will have holes in it from these prions eating away at the brain. And this actually was first discovered in sheep. So the initial form of the disease caused by a protein was called scrapie. And it was called scrapie because in sheep who are infected with this version of the prion protein, they would literally scrape all of their hair off. And it was because it was eating away at their brain, so it was causing this neurotic behavior. Another one to mention here, E. coli 0157H7. This is basically referring to the antigens or the molecules on the surface of this bacteria that really contribute to how sick you will be when you're infected with a bug. But this strain in particular of E. coli causes awful, severe diarrhea. So E. coli, normally a lot of us have a strain of E. coli in our guts, but not this strain. If you are infected with this strain, you will become sick. And it's similar to common food poisoning. And this is usually, the common name for this is traveler's diarrhea. So this is something that there are some more severe cases of this, but usually people will recover in a few days. And then we have the fun virus here, the topic Ebola, which I call it fun because it is still, I think, rather mysterious as far as how we will in the future deal with outbreaks and how we'll treat it. Currently now it's raging across West Africa, and this is actually, I think, one of my favorite pathogens to follow because we have no cure. We still don't have a great treatment for it. And it was first identified in Africa near the Congo in the last year. For the first time in our history, Ebola came to the U.S. And we actually had the first Ebola patient case transmission in Dallas, Texas. Thomas Duncan died of Ebola. It's very scary. It's a virus that if you contract, you have about 10 days to live unless you're admitted to the hospital. You basically bleed out from every orifice. It causes your immune system to go in overdrive. It causes an awful fever and internal hemorrhaging. So this is a virus that we're still learning a lot about. We don't know where it came from. There are a lot of scientists that claim that the reservoir, the natural habitat or host for this virus is a bat. And it's thought that from accidental transmission of people consuming bats in Africa for food, or also just accidental transmission from direct contact between humans and animals in the wild, that this disease may have come about, but we still have a lot to learn about it. Also, Cryptosporidium, this is caused by a protozoa that causes 30% of diarrheal illness in developing countries, and it's spread via contaminated water. So usually water, in order for water to be potable or drinkable, when it comes out of your tap, before you're actually able to drink it out of your faucet, it has to undergo all of these water safety tests to ensure that it is free of waterborne pathogens, such as this one. But there are cases where some sneak through the cracks and you have these isolated outbreaks of waterborne illnesses. All right, and lastly, we have AIDS, which stands for Acquired Immunodeficiency Syndrome. So this is kind of clinical, full-blown HIV infection, which leads to AIDS. Basically, HIV is a virus that will preferentially target the white blood cells. So it wipes out an individual's immune system and depletes them to the point where they are just riddled with all kinds of opportunistic infections. So they see infections that a healthy person would never see. And this is an infection that was first identified in the early 80s, and it's been raging across the world since then. So it is a worldwide epidemic. There's 30 million people still per year that are infected, and the reality is that we do not have the best treatment for it. There's no cure. There's no vaccine. There's still a lot of research now and work that we have to do in the future to ensure that we do find a cure for this infectious disease. And I do believe that it will happen in the near future. There's a lot of talk about novel therapies and clinical trials that are going on. So I think that there is light at the end of the tunnel as far as treating this infectious disease. Now, this is the end of lecture for week 1, so I hope that you guys got a lot out of this introductory lecture, and I hope that you also get a lot out of the whole scheme of the course. Please just, for a few heads up for the course, stay on top of the reading. Please listen to these lectures every week. They are designed to facilitate your involvement in discussions and forces you to actually hear the pronunciation of some of the microbial terms, which can be tricky at times. And it also is really a good review for everything that you'll be seeing in your textbook and also the online assignment sections as well. Now, the lecture portion, you are required to do the discussions, the online assignments, but please do not forget the lab. The lab is a huge portion of this course, so stay on top of things with the lab. Please start the experiments on time. The first week or so, I understand, takes a little while to get acclimated, so I will be flexible with that. But starting in week 2, there are no excuses. Also, I am, along with all of the students, I am conducting all of these experiments at home. So when you guys have an issue with one of these experiments, there's really no excuse that something didn't work, because if it works for me, it should work for you. So please let me know, though, if something goes wrong so I can better assist you. So I will see you guys next week for lecture in week 2.